Prophase 1. Prophase 1 starts out with a diploid cell. In the cell, the nuclear membrane disappears, and centrioles can be seen. There are also double-stranded homologous chromosomes that are being formed. Metaphase 1. In metaphase 1, the homologous chromosomes line up at the equator, forming tetrads. Spindle fibers are formed from the centrioles and attach to the centromeres of the chromosomes. Crossing over also occurs in order to mix up the genetics to increase variation. The synapsis is when the chromosomes cross over. Anaphase 1. In anaphase 1, the chromosome pairs are split apart and pulled to opposite poles of the cell by the spindle fibers. Non-disjunction can occur during this phase if the chromosomes do not separate correctly. Telophase 1. In telophase 1, the cell begins to separate. The cell pinches in at the cleavage furrow, or a cell plate will form. The nuclear membrane also begins to reform around the new pair of chromosomes in each daughter cell. Prophase 2. Prophase 2 starts out as a diploid cell. In the cell, the nuclear membrane breaks down and the centrioles are become visible. There is only one pair of chromosomes, and no replication occurs. Metaphase 2. In metaphase 2, the homologous chromosomes line up at the equator, and spindle fibers attach to their centromeres. Anaphase 2. In anaphase 2, the chromosomes are pulled apart by the spindle fibers. These spindle fibers then pull each part of the chromosome to opposite poles of the cell. Telophase 2. In telophase 2, the cell begins to pinch in at the cleavage furrow, or a cell plate will form. The nuclear membrane then forms around the sister chromatids, and 4 new diploid gametes have been formed.